Hi, this is Mr. Wartsky, and today we are going to start our journey into evolution. Remember, if anytime you need to pause the video, pause the video. If you have any questions, please refer to your textbook, or you can watch the video again in case you missed something, or you can ask me tomorrow in class. So let's get started on evolution, the consequence of genes. So evolution, and we need to define evolution because evolution is a change in gene frequencies of a population over time. It's a very narrow definition. I think we can deal with it, and we need to note that populations evolve. Individuals cannot. Populations will change, individuals can't change. So we have evidence that populations have changed over time. And you might be asking, what type of evidence do we have? And I'll be more than happy to provide you with said evidence. The first evidence of evolution are fossils. Now, fossils are evidences of once living organisms. I guess that would what organisms would be once living. So evidences of organisms. So there are different types of fossils. We have mold fossils, which are impressions, which are right here. So the little shells there made a little impression in the mud, which then lithified or formed rocks to become mold fossils. Then we have cast fossils. In cast fossils, we form the mold first, then something fills in the mold to form a cast. In permineralization, minerals fill pores of organisms, like petrified wood. It is now a rock, but initially they had wood. The pores were filled with minerals, and over time they lithified to form rock. In replacement fossils, the whole organism is eventually replaced by minerals. So we have a picture of a shelled organism here, or the shell of the organism, but the shell isn't there anymore. It's been replaced totally by minerals, and so we have a fossil. Hard parts, bones, teeth are left behind. That's pretty straightforward, I guess. And preservation. So like insects trapped in amber, mammoths trapped in glaciers. We have peat bogs and tar pits, all that can cough up the remains of once living organisms. Said it again. So those are the different evidences of evolution, and hopefully you got them all in your notes. If not, remember, hit pause. So we now need to talk about fossil dating. No, this is not dating old people. This is coming up with the date of when the fossils or the organisms were alive. So there's two types of dating. There's relative fossil dating. This is not dating your grandmother or grandfather. So what we're going to do is look at layers of sedimentation from old streams, rivers, and ponds. Older sediment layers are at the bottom. New sediment layers or younger sediment layers are at the top. They form something known as a strata or stratum, and the strata is a layer. So we compare the fossils at different strata. The lower fossils, the older they are, the higher up the fossils, the younger they are. So we have a picture of the different strata. So we have these trilobites down here, which are lower than the dinosaurs up here. So relatively speaking, the dinosaurs are younger than the trilobites. So then we have absolute fossil dating. Now this does not imply errorless. There still can be mistakes made. The first type of absolute fossil dating uses radioactive isotopes. It's called radioactive dating. You do not glow when you go on the date. Such a funny guy. So we need to know that radioactive isotopes are unstable, but they want to be stable. So what they do to become stable is they give off subatomic particles. In this process, they decay. So each radioactive isotope has a fixed rate of decay in order for it to become stable. Now, each rate of decay is expressed in half-lives. The half-life is the amount of time it takes for a half of the isotope to become stable. So for example, carbon-14, which is a radioactive isotope, has the half-life of about 5,600 years. Now, all organisms take in and give off radioactive isotopes. However, we usually have a consistent amount of radioactive isotopes in our bodies at all times. But when we die, we stop taking in the radioactive isotopes and they will start to decay. When we compare how much we have with how much we should have with the rate of decay, then we can come up with an approximate time when we died and therefore when we lived. Now, radioactive isotopes is not affected by temperature and pressure. So we have radioactive isotopes here. The half-life it takes for half of them to become stable right here is about 1 billion years. So we have half, and then we have another half, and then we have another half, right? So then we can compare parent isotope to radioactive isotope. It decreases over time. So this is 1 half-life, this is 2 half-lives, this is 3 half-lives. So if we find the amount of radioactive isotopes that are present compared to the amount that should be there, let's say there's 1/8, that means that it's gone through 3 half-lives. And then we can approximately tell when that organism died. Hopefully that made sense. The second type of absolute dating is called amino acid racemization. And amino acids come in 2 isomers, left and right. L stands for left, obviously, and D stands for right. I guess that's French word for right. We use and produce left-handed amino acids. If we're going to abbreviate that, that would be LA, left-handed amino acids. And when you die, your left-handed amino acids change to right-handed amino acids, which is DAA or DA. So that's right, it goes from LA to DA, right? Left-handed to right-handed amino acids. If the rate of change is consistent and we can compare the left-handed to right-handed amino acids, we can estimate the time of death. But of course, we need to have proteins around so we can compare the amino acid isomers. This is affected by temperature, though. So here on the left is our left-handed amino acids, right, our right-handed amino acids, and they are different. So you go from La to Da. Another type of evidence, and we've only talked about fossils and fossil dating so far as evidence of evolution. Another form of evidence, we are going to compare anatomical parts of different species. And when we compare them, we're going to look for homologous parts. So what are homologous parts? Well, they are parts that look similar but have different functions. Organisms that have homologous parts, they had a common ancestor but were exposed to different environmental conditions. And so they had different solutions to different problems. If you take a look at the forelimbs, the arm of all these different organisms here, you will notice that the bones are arranged in a similar order and they are similar bones. We're going to do something like this in class. Those are homologous parts. They look similar, but they do different functions. They convey a common ancestor and the populations are exposed to different environments, so different solutions to different problems. We can compare anatomical parts and look for analogous parts. Now, they look different, but have the same function. So this shows you that they don't have a common ancestor, but yet they've been exposed to the same environmental problems. So they have similar solutions to the same problem. And these are wings of a moth. I don't know what that is, but we have a bird and a bat here, and they are all different. This might be a pterodactyl. I'm hoping. So they're all analogous because they do the same thing, but they are totally different. For example, moths don't have bones and feathers or skin. This has skin, this has feathers, this has skin. This is the hand. These things are the fingers. So although they solve the same problems, different common ancestor, analogous parts. Sometimes when we take a look at anatomical parts, we find vestigial parts which have no apparent function. Now we need to be careful about this because just because it doesn't have a function, since the structure there doesn't imply that it once had a function. It might have, like the vestigial hips in snakes and whales, but not necessarily. The only function that it has is that it does not kill you. And so some vestigial structures for us would be our eyebrows. They think that the appendix does have a function, that it harbors bacteria or holds bacteria for situations where you need to repopulate your colon. That could be a function right there. So we have a lot of like our tailbones, but just because we have them, that doesn't mean that it had to have a function. The only function that it has is it doesn't kill you. And then we have comparative biochemistry. That's another form of evidence of evolution. In comparative biochemistry, we are comparing proteins or amino acids or DNA of different organisms to see if those things are similar. Organisms that have similar proteins, similar amino acids, or similar DNA have shared a common ancestor. And biogeography. We need to take a look at the distribution of the organisms. Organisms don't usually travel that far. If they are similar looking and they are close by, there's a good chance that they shared a common ancestor. Just like Darwin's finches. There were 13 of them. There's one population that came over from the mainland, and on the different islands, they're close together, they had different finches. Biogeography. And then finally, looking at the developing embryos, comparative embryology. Now, they used to say if they developed similarly, then they shared a common ancestor. But this has been proven false because the person who came up with this hand drew the pictures and kind of fudged the results. But the College Board is a little slow, so if they talk about the evidence of evolution, this might be listed as an evidence. But you and I, we know that it's not. So how did the populations change? Well, there are a couple theories based on the individuals who proposed them. The first one that we're going to talk about is Jean-Baptiste Pierre-Antoine de Monet Chevalier de Lamarck. For us, it's going to be Jean-Baptiste de Lamarck or Lamarck, because we're American. Now, he had a couple of theories of how populations changed. His first idea was called the law of acquired characteristics, and said that organisms can change their bodies when they needed to and then pass on those changes to their offspring. So there's an example of a giraffe that all giraffes had short necks, and then what happened was in order to eat the leaves off of trees, they stretched their necks until all giraffes had long necks, and they passed those long necks on to their offspring. Now, also, if you got in a terrible accident and lost an arm, and then you had kids, your kids would not have an arm. Acquired characteristics, right? Now, we laugh at him, but we are constantly Lamarckian. Like polar bears are white because they live up north. Well, no, they're white because the ones that were white survived and the other ones died. All right. And there are other examples of how we are Lamarckian. We cannot be Lamarckian. His second law was the law of use and disuse. He says if you don't use it, you will lose it. Same thing with the appendix. So we think that since we have these structures, they must have had some type of importance, because if they did not have a function, then we wouldn't have them. And that's not true. And Lamarck wasn't true. Okay? And we're going to talk about this later, but our imperfections is an evidence of evolution. So perfection would be evidence of a creator. But that's where we're going to stop right here. We're going to talk about Darwin in the next screencast. And have a good night.